Imagine you're trying to figure out how a really bustling, chaotic city works. Like, you want to know the traffic patterns, how different neighborhoods interact, where the industrial zones kind of bleed right into the residential blocks.
Right.
And if you only look at satellite images from space, I mean, you can see the broad strokes, you see where the gray concrete ends and the green parks begin. But if you want to know what's actually driving the local economy or, who lives in a specific house, that satellite view is practically useless. You need to zoom all the way down to the pavement. You really need that street view.
Yeah, exactly. And the biology that we desperately need to understand to cure diseases, it's happening entirely down on that street level.
Right.
Yet the whole field of computational pathology has been somewhat, well, stuck in satellite mode. The big picture is there, but the fundamental unit of life, like the individual cell, it just gets blurred out in the aggregate.
Totally. Well, welcome to today's deep dive. We are super thrilled you're joining us because today we are zooming all the way in.
We really are.
Our mission today is to unpack this fascinating new research preprint from a team at Institut Curie and Mainz Paris PSL. Specifically, we're looking at research led by Loic Charuteau, Alice Blondell, and their team.
Yeah, a really incredible group.
The paper is titled lemon, a foundation model for nuclear morphology in computational pathology. And we're going to break down how this brand new AI model is basically learning to look at microscopic single cells the exact same way a world-class pathologist does.
Which is wild to think about.
It is, and doing it at a scale that could totally revolutionize cancer research and precision medicine for, everyone. So, okay, let's unpack this. Before we get into how this lemon model actually works, we really have to understand the roadblock.
Right, the bottleneck.
Exactly. Like, why is looking at a tissue sample such a massive computational headache to begin with?
Well, it really just comes down to sheer overwhelming scale. When you go to a hospital and a biopsy is taken, that tissue is put on a little glass slide and scanned into what we call a whole slide image, or the WSI. And these digital files are just gigantic. A single digitized slide can easily be more than 50,000 pixels wide.
50,000 pixels. Oh, wow.
Yeah, I mean, if you printed that out at standard resolution, you'd basically have a picture the size of the tennis court.
That is insane for one slide.
For one slide. And typically, these tissues are prepped using hematoxylin and eosin staining. We just call it H&E in the field.
Right, H&E.
It's this chemical dye process that reacts with the tissue.
Yeah.
And it turns the nuclei, this dark, crisp purple, and the surrounding structures a softer pink.
Which, if you are listening to this and you ever look through a microscope in, like, high school biology, you've probably seen that exact pink and purple landscape.
Exactly. It's an incredibly common, very cheap process that's been used for over a century now.
And doctors are looking at this landscape to figure out what's happening in the tumor microenvironment, right? Like, they are trying to see the battlefield, how the cancer cells are physically interacting with the patient's immune cells and the structural tissue.
Yeah, that's the goal. But that battlefield is made up of billions of individual cells across a single slide.
Right.
Having a human pathologist manually analyze every single one of those cells, figure out its exact type, and map its relationship to its neighbors, it's biologically impossible. It would take a lifetime.
Right, there's just not enough time in the day. But don't we have, like, advanced molecular techniques today? Things like spatial transcriptomics?
We do, yeah.
Yeah.
Those can map out the exact gene expression like the molecular machinery running inside the cell.
Right.
But the problem is those techniques require destroying the tissue. They're incredibly expensive, and they can only really be run on tiny fragments of data.
Ah, so it's not a practical, everyday solution.
No, not at all. So the medical field naturally turned to artificial intelligence to analyze those cheap, abundant, tennis court-sized H&E slides.
Enter the foundation models. These are the AIs trained on just massive amounts of data.
Exactly.
But if I'm reading this preprint correctly, the way they've been doing it so far has a pretty massive blind spot, right?
Yeah, it really does. Most of the current heavy-hitting AI models in pathology, these are massive models with names like UNNI or Virtue2 or H-Optimus0. They use self-supervised learning, but they apply it to tiles or patches.
Okay, so what does that mean exactly?
It means they take that 50,000 pixel wide slide and they basically chop it up into large, arbitrary chunks of tissue, usually like 224 by 224 pixels.
Which brings us right back to that satellite imagery analogy.
Exactly.
The AI is looking at the overall texture of a neighborhood of cells. It can say, oh, this looks like a dense urban area, or this looks like a suburb.
Right. It gets the gist.
But it is completely missing the single cell level. Like, it can't see the specific features of the nucleus, which is absolutely crucial for characterizing specific cell types.
Exactly.
But that raises an obvious question for me. If looking at individual cells is so vital to understanding cancer, why hasn't it been done at scale before?
Because extracting and organizing millions of individual cells from those massive 50,000 pixel wide slides is computationally brutal. You are basically searching for microscopic needles in a massive digital haystack.
Right, I can imagine.
But that is exactly what the team behind Lemon managed to solve.
And just to clarify, Lemon stands for Learning Embeddings from Morphology of Nuclei, right?
You got it.
Yeah.
So to build it, the researchers utilized this highly optimized segmentation tool called Hoverfast. You can kind of think of hoverfast as an ultra-fast automated microscope.
Okay.
It scans through these massive slides, identifies the precise boundaries of every single nucleus, and physically cuts them out one by one in the digital space.
And they didn't just do this for a handful of slides either. I mean, they went into the TCGA database, the Cancer Genome Atlas, and ran this automated extraction across about 10,000 whole slide images.
Yeah, it's spanning 10 completely different cancer types, too.
That's a ton of data.
Yeah, the resulting data set is just staggering. They extracted millions of single-cell images. But the critical detail, the thing that changes the entire math of the AI training here, is the size of these new images.
They're small, right?
They are tiny. They are just 40 by 40 pixels, perfectly centered on a single nucleus. We're talking about 10 micrometers across.
Wow.
Yeah, we have literally gone from a 50,000 pixel wide continent down to a 40 pixel wide house.
Okay, so we have millions of tiny 40 by 40 pixel images of cells, but it feels like we just traded one problem for another here.
How so?
Well, how do we actually teach an AI to understand them? Nobody is sitting in a lab manually labeling millions of tiny pink and purple blobs saying, oh, this is a lymphocyte, and this is a fibroblast.
Right, that would take lifetimes.
How do they do it?
That is where the concept of self-supervised learning, or SSL, comes in. In traditional AI training, you give the AI an image and a label, like an answer key.
Like saying, here's a picture of a cat, and the label says cat.
Exactly.
Yeah.
But in SSL, you throw away the answer key. You literally force the AI to find the underlying structure of the data all by itself by playing this really complex game of comparison.
Okay, interesting.
The researchers actually tested the two leading frameworks for doing this to see which one could handle these incredibly tiny images, MoCuV3 and DINOv2.
Okay, let's pause there, because those two frameworks represent a pretty massive clash of AI philosophies, from what I understand. Starting with MoCuV3. The paper says it uses contrastive learning and something called the InfoNCE loss function.
Yeah.
How does that actually work in, like, plain English?
Okay, imagine you are sorting a massive pile of unlabeled photos in the dark.
Okay, I'm with you.
In contrastive learning, you take an image of a cell and you apply two different random visual distortions to it. Maybe you rotate one version and you change the color grading of the other.
Like putting an Instagram filter on one of them.
Sure, exactly.
Yeah.
So you show both of these distorted views to the AI and say, hey, these are the exact same cell, pull their mathematical representations closer together. Then you show a completely different cell from the pile and say, this is different, push them apart. You are constantly contrasting positive pairs against negative pairs. That push and pull mechanism, that's the InfoNC loss function at work.
Okay, pull similar things together, push different things apart. It builds a map based on similarities. That makes complete sense. But then there's DINOv2, and this one is non-contrastive learning.
Right, DINOv2 completely discards the idea of negative pairs. It doesn't push different things apart at all.
Wait, really? Then how does it learn?
Instead, it uses a teacher-student network approach. But the crucial mechanism is something called local-to-global matching. The AI takes a small local crop of an image and tries to predict the broader global context of the full image.
Wait, I'm confused, because DINOv2 is famous for being absolute state-of-the-art for natural images, right? It's what powers some of the most advanced computer vision systems out there.
It is, yeah.
So why would an older, or I guess simpler method like MoCuV3 beat it in this experiment?
Well, what's fascinating here is how an AI architecture designed for high-resolution photos of dogs and cars completely breaks down when you apply it to microscopic biology.
Oh, because of the size.
Exactly. Think about that local-to-global matching in DINOv2. If you have a high-resolution photo of a dog, a local crop might be, the dog's ear or its paw.
Right.
And the AI can look at that highly detailed ear and deduce, okay, the texture of this fur and the shape of this cartilage means this is probably attached to a dog.
Makes sense.
But in our pathology case, the global image is a single cell that is only 40 by 40 pixels wide.
Oh, I see the problem. A local crop of a 40-pixel image is what, like 10 pixels?
Basically, yeah.
So a local crop of a dog is an ear, but a local crop of a 40-pixel cell nucleus isn't an ear. It's just like 3 purple pixels.
Yes.
You're asking the AI to look at a single purple paint chip and guess the entire painting.
Exactly. It is mathematically ill-posed. There is absolutely zero useful biological information in a 10-pixel crop of a nucleus. It's just a blurry purple gradient. The researchers actually had to surgically remove that local-to-global matching component from the DINO loss function just to get the program to run without crashing.
That is wild. And even with that modification, DINOv2 had another, catastrophic failure point, right?
Yeah.
The paper mentions something called the 𝗏 regularizer. What is that, and why did it break?
Right. So remember, the DINOv2 doesn't use negative pairs to push different images apart.
Right, no pushing.
Because of that, the AI can sometimes take a really lazy shortcut. It realizes, hey, if I just guess that every single image is exactly the same, I technically never get the answer wrong.
Oh, tricky.
Yeah, so to prevent that collapse, DINOv2 uses the 𝗏 regularizer. It's basically a mathematical rule that forcibly shoves the representations of the images apart, kind of scattering them across the AI's internal map.
But wait, forcing things apart only makes sense if the things are actually different?
Exactly.
If I give you 1,040,000 images that are all just slight variations of a tiny purple blob, and I mathematically force you to find extreme differences between them...
The math literally rips itself apart.
Oh, man.
Yeah, the regularizer was way too aggressive for these tiny, visually similar cell patches. It pushed the data points so far apart, so violently, that it was causing numerical errors during training. Like, the numbers literally became too extreme for the system to compute.
Wow. So what did they do?
The team had to replace that aggressive rule with a much milder, custom kernel density estimation loss. They call it a KDE-based loss.
So even after performing major surgery on Dino V2, like removing the local to global matching and totally swapping out the regularizer, the simpler Markov3 still won out.
It did. The global contrastive objective of just looking at the whole 40 by 40 patch and saying, pull this together, push that apart, was just fundamentally better suited for the reality of microscopic pathology.
It's like you said, the architecture really has to match the reality of the data.
Precisely.
Which brings us to a massive hurdle. We've established that AI knows how to learn using Markov3. But training an AI on pristine controlled data in a lab is one thing. If you want this tool to actually work in a real hospital, it has to survive the chaotic, messy reality of thousands of different laboratories, right?
Absolutely.
How do they prepare the AI for that?
This is where domain-specific augmentations become critical. When you train an AI, you apply random artificial distortions to the training images. You're essentially hazing the AI, making its job artificially harder, so that when it sees clean data, it's incredibly robust.
Like the Instagram filters we talked about.
Right. With a natural image, like a car, you might flip the picture horizontally to teach the AI that a car pointing left is the same as a car pointing right.
Yeah.
But a cell isn't a car, it's floating in tissue. There is no right side up. So the researchers applied a full 360-degree rotation augmentation.
Okay, but wait, rotating a square image creates a geometry problem.
It does.
Like if you take a 40 by 40 square and rotate it 45 degrees, the corners of the original square rotate inward, and you are left with these empty black triangles in the corners of your new image frame. Yes. If you feed that to an AI, it's not going to look at the cell. It's going to obsess over those stark, fake black edges, right?
Absolutely. AI models are notoriously lazy. They'll latch onto any artifact they can find. To bypass that, the team used a brilliant, highly specific workaround.
Okay.
They went back to the original massive tissue slide. Instead of cutting out a 40 by 40 square, they cut out a larger 60 by 60 square.
Oh, I see where this is going.
Yeah, they took that larger square, rotated it a full 360 degrees, and then cropped the central 40 by 40 pixels out of it.
Presto.
Exactly. Perfect. Seamless rotation with zero black edges.
Yeah.
The AI only sees continuous biological tissue.
That is so elegant. Just so smart.
It really is.
But orientation is only one variable. The much bigger issue in pathology is color, right? We talked about those pink and purple H&E stains earlier. From what I understand, they're notoriously inconsistent.
Oh, wildly inconsistent.
Like a lab in New York might use a chemical dye that produces a deep, rich magenta, while a lab in Tokyo might use a slightly different manufacturer, resulting in a faded, pale pink.
Right. And if the AI learns to rely on the exact shade of pink to identify a cell, it will completely fail the moment you hand it a slide from a different hospital.
That would be a disaster.
So to prevent this, they used a technique called Marchenko stain normalization, combined with heavy grayscale and color jitter augmentations. They are constantly shifting the hues, the saturation, and the contrast during training.
It's sort of like teaching someone to become a master sommelier.
Okay, how so?
Well, you want them to be able to recognize the exact vintage and grape of a wine, even if you serve it to them in like a coffee mug or a red plastic cup or under flickering neon lighting.
Huh, yeah.
You're forcing the AI to ignore the presentation and focus entirely on the taste, the actual underlying biological structure of the chromatin folding inside the nucleus. You're forcing it to ignore the specific dye it's served in.
That's a great analogy. And because the researchers were so meticulous about isolating that specific structural taste, they didn't need a massive, power-hungry AI architecture. They actually used a vision transformer, specifically a VITS-8 model.
Let's unpack that term really quick, because when most people hear transformer, they think of ChatGPT predicting the next word in a sentence. How does a text prediction brain look at a visual cell?
It's actually a very similar concept. Instead of breaking a paragraph down into words or tokens, a vision transformer breaks an image down into tiny visual patches.
Okay.
It treats those visual patches like words in a sentence, and it learns the relationships between them. And because the Lemon model is only looking at a 40 by 40 pixel image, the number of words it has to process is incredibly small.
Well, here's where it gets really interesting. Because of that small, focused architecture, the computational efficiency of this model is just mind-blowing.
It really is.
We measure AI processing power in FLOPS floating point operations. It's essentially how much math the computer has to do to process one input.
Right, and to process a single 40 by 40 image, Lemon requires just 0.5G FLOPs.
Half a gigaflop.
Half a gigaflop. Contrast that with ℏ_0, one of those massive tile-level models we discussed at the start, which looks at 224 by 224 pixel chunks. That model requires 295.9 G FLOPs per image.
0.5 versus nearly 296. That is hundreds of times cheaper to run.
Exactly.
When you consider that a single slide has millions of cells and a clinical study might have thousands of slides, Lemon takes a process that would basically require a multi-million-dollar supercomputer.
And turns it into something you could realistically run on a standard hospital server.
That's huge.
It completely lowers the barrier to entry for computational pathology.
But, an AI can be fast and lightweight all day long. None of that matters if it doesn't actually understand the biology better than the heavyweights.
True.
So when they finally took Lemon out of the lab and put it to the test on unseen data, what actually happened?
They put it through a rigorous set of benchmarks using a process called linear probing.
Linear probing.
Yeah, essentially, they freeze the AI's internal brain so it can't learn anything new. Then, they attach a simple sorting algorithm to the end of it and test how well the AI's internal representations can separate different types of cells.
Okay, the final exam.
Yeah. First, it completely crushed models trained on natural images, like ImageNet.
Which proves that training an AI on millions of photos of cats, dogs, and bicycles does not teach it how to understand the morphology of a nucleus.
Right.
That makes sense. But what about the massive tile-level models? The ones burning nearly 300 billion FLOPs?
Lemon beat them too.
Wow.
Yeah, they tested it on a dataset called 𝗦𝗁𝗠𝗡, which asks the AI to identify atypical versus normal mitotic figures. Essentially, the AI has to catch cells in the exact act of dividing abnormally, which is a key indicator of aggressive cancer.
Right.
Lemon outperformed 𝗦𝗁𝗠𝗏, 𝗦𝗁𝗠𝗏, and 𝗏_𝗏. It also beat them on the 𝗚CLS dataset, which is a highly complex breast cancer cell classification task.
It even outperformed Volta, right, which was previously the only other dedicated cell-level model out there.
Yep, it beat Volta as well.
So the lightweight, highly focused model wins the spreadsheet war. But the truly mind-bending part of this paper isn't just that it scored higher on a test. It's how the researchers proved what the AI was actually learning.
Yes.
We really need to talk about the latent space.
The latent space. So the latent space is basically the AI's internal map of the world. Every time the AI looks at a cell image, it assigns that cell a specific coordinate in a massive, multidimensional mathematical space.
But human brains can't comprehend a map with hundreds of dimensions. We get lost in 3.
Exactly. So the researchers used a mathematical technique called a 𝗬 projection.
How does that actually work? How do you visualize something that complex?
Imagine trying to draw a 3D solar system on a flat piece of paper. You have to squash the depths and distances while trying to keep the relative positions as accurate as possible, right?
Right, like drawing a map of the globe.
Exactly. A 𝗬 projection does that for the AI's brain. It flattens that complex multidimensional map down to a 2D image, so human researchers can actually look at how the AI is grouping the data.
And remember, because this was trained using self-supervised learning, the AI was never explicitly taught the rules of biology. Nobody ever told it what a cancer cell is or what an immune cell is.
Not once.
It was just told to pull similar shapes together and push different shapes apart.
Yet, when you look at the flattened 𝗬 projection of Lemon's latent space, the AI has naturally, organically rediscovered the biological taxonomy of cells.
That is just incredible.
It put all the epithelial cells over on the right side of the map. It gathered the small, round lymphocytes into a tight, distinct cluster. It pushed the elongated, spindle-shaped fibroblasts all the way to the bottom.
Oh.
Without a single label, just by looking at the morphology, the physical shape, and texture of the nucleus, the AI figured out the distinct categories of life.
I mean, I understand how looking at the shape of a cell can tell you what kind of cell it is. But the researchers went a massive step further here. How can an AI that only looks at the shape of a cell predict something completely invisible, like gene expression? Because knowing what a cell is doing on a molecular level, that is the holy grail of cancer research.
It really is. And to test this, the researchers used the ECEST dataset, which contains spatial transcriptomics data.
Okay, break that down for me.
In this dataset, we know the exact ground truth. We know precisely which genes are turned on or off in a specific cell. The researchers took that invisible genetic data and laid it over Lemon's morphological map. The correlation was striking. The physical shape of the nucleus perfectly predicted the expression of well-known marker genes.
Wait, really?
Yes. Transcriptionally related cells, cells that were running the exact same molecular programs, occupied the exact same neighborhoods in the AI's visual mind.
I want to make sure I'm visualizing this correctly. It's basically like looking at the exterior architecture of a person's house, like the slope of the roof, the size of the windows, and using nothing but that outside shape to perfectly predict their grocery list inside the refrigerator.
That is exactly what is happening. The physical folding of the chromatin, the density of the nucleus, those visible features are the direct result of the invisible genetic machinery running inside. Lemon learned to read the exterior architecture so perfectly that it can deduce the molecular grocery list.
So what does this all mean? Like, why should you, listening to this, care about a small vision transformer looking at 40 pixel squares of purple dye?
Well,
You should care because this gives doctors, researchers, and ultimately patients a massive, incredibly cheap shortcut to understanding a specific cancer.
Exactly.
If we can reliably predict molecular function and precise cell types just from the physical shape of the nucleus on a cheap century-old H&E slide, we can totally bypass the need for millions of dollars in manual labeling.
We can skip the incredibly expensive tissue-destroying molecular sequencing entirely.
Right. We can take the highest level of precision medicine and democratize it, making it available to literally any hospital with a standard computer server and a digital scanner.
It fundamentally changes the economics and the accessibility of computational pathology. We are extracting entirely new layers of biological truth from data we already possess.
It's just brilliant. Let's quickly recap the journey we just went on. We started with massive tissue slides that were 50,000 pixels wide.
Huge.
We realized that the big AI models were missing the microscopic details by only looking at large neighborhood-sized tiles. So the researchers zoomed all the way in, utilizing hoverfast to extract millions of tiny 40 by 40 pixel single-cell images.
Right.
They discovered that complex AI frameworks like DANOV2 mathematically ripped themselves apart at that tiny scale, so they adapted Markov 3's simpler contrastive learning.
Because simpler was better.
Exactly. They threw in brilliant augmentations, like the 60 by 60 crop trick to handle ropation without black edges, and they normalized the stains to ensure the AI focused on the structure, not the dye.
The wine, not the glass.
The wine, not the glass. And they ended up with Lemon, an incredibly efficient model that uses a fraction of the compute power, but naturally understands the invisible language of biology, linking the shape of a nucleus directly to its internal gene expression.
If we connect this to the bigger picture, there's a detail tucked into the conclusion of this paper that is absolutely thrilling.
Oh, lay it on me.
To build Lemon, the researchers deliberately excluded the spatial distribution of the cells. They isolated just the single-cell morphology. They entirely divorced the cell from its neighbors, ignoring the architecture of the tissue to force the AI to look at the nucleus.
Right, they took the street view, but they basically hid the rest of the city map.
Exactly. But here is the provocative thought I want you to mull over.
What happens when we finally bridge the gap?
What happens when we take Lemon's perfect, intimate street view of individual cells and we combine it with a macro neighborhood view of the massive tile-level models, the models that see how those cells are physically arranged next to each other?
That would be the ultimate map.
Right. Could that multi-scale integration finally unlock the complete spatial syntax of a tumor? Could we see not just who lives in the house, but how they are communicating with the neighborhood to help the tumor breathe, adapt, and grow?
Now that is the kind of question that keeps the scientific community up at night, because once you can read the granular details of the streets and you understand the flow of the whole city at the exact same time, you can finally start to rewrite the rules of the traffic.
Exactly.
Thank you so much for joining us on this deep dive into the sources today. We hope it gave you your own aha moments. Keep questioning, keep zooming in, and keep exploring the invisible worlds around you. Until next time.